Hi everybody, this is Unit 10, the Cardiovascular System. It is one of the longer PowerPoints of this class, so there will be general stopping points, and I'll let you know when those are if you want to stop this PowerPoint and come back later. So the cardiovascular system includes basically a pump, tubes, and blood, the liquid. And we're going to talk about all three, starting with the components of the blood and how blood is made. Then we'll talk about how that blood travels through the tubes or the vessels. We'll talk about the heart, the pump itself, and then how to control things like nutrient exchange at the capillary levels, blood pressure, cardiac output, and some techniques used to evaluate heart function. Heart disease is the #1 killer in the United States and worldwide, so there's a lot of information about this and a lot of research into it to help people control their blood pressure, for example. So again, the components of the cardiovascular system include the pump, which we call the heart, the blood vessels, arteries, veins, and capillaries, and the blood itself, the liquid that is circulating through those vessels. So the functions of blood are to transport nutrients and gases to the tissues, help regulate pH, perform clotting when there's an injury to a vessel, defend us against incoming pathogens, and stabilize temperature. It takes a lot to change the temperature of water, and we have over 5 liters of fluid just in our blood alone. And if it's all at 98.6, it takes a lot to change that temperature. So having that much blood is actually good. It helps stabilize our temperature. Okay, so the average liter has about 5 liters. Think about a 2-liter bottle. We have 2.5 liters of that. More if you're a bigger person, less if you're smaller, obviously. Men have slightly more blood than women in general. And each of those milliliters, so basically in a teaspoon, I think there's 5 mils in a teaspoon. If you had a teaspoon of blood, and each one of those milliliters has 5 million red blood cells, you'd have 25 million just in 1 teaspoon of red blood cells. So you have a lot of red blood cells in your blood. And that's good because the function of a red blood cell is to carry oxygen. And without oxygen, you don't live very long. When it comes to white blood cells, you have 5 to 10,000 of those per mil, so a lot of those too. And of course you also have platelets, which we'll talk about that in a few minutes. But if you take your blood and you put a little tiny bit in a tiny capillary tube and spin it down with a centrifuge, that's called doing a hematocrit, or that is a hematocrit, and there's a picture of one here. You can see that the red blood cells will settle at the bottom. That little white stripe between the red and the plasma, the yellow part there, is the buffy coat where the white blood cells are, and then the rest is the plasma. And if you measure the distance of the red and divide it by the total distance, that gives you your percent hematocrit, or your percent packed cell volume. And it should average about 45%. Up to 52 if you're a man, and down to 36 if you're a woman. But if it's below that, you don't have enough red blood cells and you would be diagnosed as anemic. Okay, so this is something you might do in lab someday. I think we do it in A&P2 if you come to class. It's not something we can send home in lab kits because you don't have a centrifuge at home to do it. So besides the red blood cells, the plasma, that straw-colored fluid at the top in the hematocrit, is what the blood cells are floating around in. But you also have white blood cells and platelets in the plasma. The red blood cells make it look red, okay, which is why in this picture here, when you spin it out, the plasma looks kind of a straw color because you've spun out the red blood cells. Okay, white blood cells do not have any color, and platelets are actually cell fragments, and they're going to be involved in the clotting process. So what's in plasma besides blood cells? It's mostly water. There's also some proteins in there called the large plasma proteins, which helps us maintain osmolarity. And anything else that you might have eaten today, and therefore there's going to be some sugar and some amino acids, and this is going to vary depending on your diet. There's also going to be oxygen and carbon dioxide because you're always carrying gases back and forth between the tissues and the lungs. Electrolytes, particularly sodium, potassium, calcium. Any waste that your body might be generating and sending to the kidneys, you're going to find that in the blood as well. You can actually donate plasma. They'll pay you money for it. You can donate twice a week because they basically take your blood out, spin it down, keep the plasma, resuspend your blood cells in saline solution, and put it back into you. So you don't lose any volume. You have to have a needle going out and a needle going in, so maybe not as comfortable, but scientists use some of these nutrients and hormones and some of the other stuff to do various tests and analysis. So you can get paid to donate your plasma. Here's a picture I totally robbed from a copyrighted site. It's an electron micrograph, and they colored the red blood cells red. The green one there is a white blood cell, and the little yellow things are platelets. So you can see, kind of compare the size of the different blood cells in this picture. So we'll talk about red blood cells first. They're otherwise known as erythrocytes, and their function is to carry oxygen. And again, you have 5 million of them per mil, so you have a lot of red blood cells. Men have more than women in general, and you can make more. Obviously, you make these every day by the bone in your red bone marrow, but the hormone used to induce this, you can actually inject and make more. Some people abuse that. That's what the biker guy, Lance Armstrong, was doing. He was injecting himself with erythropoietin, which made him make more red blood cells so he could carry more oxygen. They're shaped a little bit like donuts that the hole doesn't go all the way through. They tend to be colored redder when they're bound with oxygen because of the iron, and then a darker color without oxygen. And they only survive about 120 days. Because they're being pushed through very tiny vessels, they get literally worn out. So you make millions of these every second, and you degrade them every day as well. Your liver and spleen get rid of old red blood cells. So here's a picture of red blood cells going through the vessels, and people who have sickle cell anemia, they have a mutation in their hemoglobin gene, and so their cells form this C-shaped or sickle shape instead of the little donuts, and they're more likely to get caught. And so the sickle cells will block blood flow, and any tissue beyond that point now isn't going to get oxygen. It can be very painful. And just in the last couple of weeks, they have announced a gene therapy treatment to treat people who have sickle cell anemia. So that's pretty exciting to be able to cure that disease after they're born, after they have it, by changing the gene in a living person. Erythrocytes also have no nuclei or mitochondria in them. They're made with a nucleus, but within a day after maturing, after they're released from the bone marrow, the nucleus goes away. The mitochondria go away. They want to make room inside that hemoglobin for as much hemoglobin as possible. And the average red blood cell will actually hold 280 million molecules of hemoglobin, each of which can bind 4 oxygen molecules. Okay, so 280 million times 4 we're talking billions of oxygen molecules per red blood cell, and we need 5 million per mil to have enough to carry enough oxygen to keep our tissues happy and healthy. Okay, so no nuclei or mitochondria because they're packed full of hemoglobin. White blood cells, also called leukocytes, there are 5 types: neutrophils, eosinophils, basophils, monocytes, and lymphocytes. What you should remember from this is neutrophils are the most common, and of the 10,000 you have, 6,000 of them should be neutrophils. The other ones are not as prevalent, but they're just as, all of them are important. They all help fight off infection. Basophils go up when you have allergic reactions because they release histamine and heparin. And so you take Benadryl, an antihistamine, to counteract the side effects of that one. We'll talk a little bit more about monocytes and lymphocytes when we get to the immune system. They're involved in this specific immune response. But all of these cells can do phagocytosis, which means they can engulf bacteria or viruses or cells that are broken and gobble them up. And again, here's some of the more specific functions of these. You should remember that you have the most number of neutrophils. And people who don't have enough neutrophils can't fight off infection. They will be, if they're in the hospital, you can't go in their room without wearing gloves and a gown. You can't deliver fresh flowers to them. Fresh fruits and vegetables are off limits because they could be covered with bacteria. So if you don't have enough neutrophils, you can catch infections very easily, and so it's kind of dangerous. Eosinophils go up when you have a parasite. Basophils release histamine. They're the ones that are involved in the allergic reactions. And then again, monocytes and lymphocytes involved in the specific immune response. Platelets, now these little tiny things here between the red blood cell and the white blood cell. They're actually fragments of cells. They only last about 10 days, and they play a big role in clotting. Okay? If you don't have enough platelets, you could bleed to death because you can't clot. So where are blood cells made? Blood cell making is called hematopoiesis, and it happens, you guys should remember, from Unit 6, in the red bone marrow, the spongy bone. Remember, at the ends of the long bones of the epiphyses have spongy bone, and that's where the red bone marrow is. And that gives rise to red blood cells, white blood cells, and platelets. Probably the most made is the reds because they degrade so quickly. Okay? So they're made by the millions every single second. And you can induce more by having low oxygen. Some athletes go up to high mountains or high altitude training centers because there's less oxygen up there. And so while they're up there, their red blood cell synthesis increases. And so they'll go up there, and when they come down off the mountain, they'll have more red blood cells than average. And they last 120 days. So if you go up there within 120 days of your athletic event, you'll have more red blood cells. You can carry more oxygen, so you have a little more endurance. One thing that induces white blood cell synthesis is having an infection, which, no kidding, that's what you need to fight off infection, right? More white blood cells. Again, erythrocytes only last about 120 days, so they're made all the time, and therefore you need all the stuff to make them. Number one, you need iron, and the most common reason people don't make enough red blood cells is they don't have enough iron. But you also need folic acid and vitamin B12. Some people can't absorb vitamin B12 very well. Vitamin B12 is also only found in animal products. So naturally it's only found in milk and meat, and if you're vegetarian, you might be low in vitamin B12. But you can take a supplement. It's fortified in some foods. Folic acid is found in green leafy vegetables, so you need to eat a variety of food to make sure you have the stuff to make red blood cells. On the other hand, you got to get rid of the old ones. The iron is pulled off and reused. We do recycle the iron. The liver does that to try to save it because, again, that's the number one reason for people being anemic, is they don't have enough iron. The remaining parts are excreted in the feces, and because of the oxidation of various of the heme, what's left over from it, that's why your poop is brown. I'm sure you came to class today, Jill, to learn why the color of your poop is brown. But there you go, it's because of the recycling process of red blood cells. As you can see that a little bit in this picture, the red bone marrow is making blood cells after they get worn out at 120 days, going through the spleen or the liver recycles it, and the proteins and the globin are broken down to amino acids. The iron is recycled. It goes back up to the bone marrow. Okay, the iron is transported in the blood by a protein called transferrin and stored in the liver. That's another reason they tell you if you're anemic, eat liver, because there's a lot of iron stored there. It's kind of a nasty way to get iron. I don't like liver, but it's a good way to get it. Alright, the last thing we're going to talk about regarding the blood is how to stop bleeding. The word for stopping bleeding is called hemostasis. Don't get this confused with homeostasis. Hemostasis, stasis is stop, right, or steady. You want to stop bleeding when you're injured. Why? Because you don't want to lose your blood, right? Too little blood will drop your blood pressure and you can't get nutrients to tissues. You could die. The other thing is there's a hole. Okay, and if there's a hole to the outside, not only are you losing blood, but bacteria, viruses, or pathogens could get in. So that's another reason to stop the bleeding, to prevent an infection. Also, you need to stop the bleeding to set up the repair system. Just like your pipes, if they burst, you're going to go turn the water off before you're going to start to fix the pipes, right? You need to stop the bleeding or stop the problem so you can set up the repair system. So how does a clotting system work? Basically, there's 3 steps, and there's a lot more to each one of these steps here. You get into a little bit more detail when you take A&P2, but essentially the first thing is your blood vessel will constrict. Okay, as soon as you break a vessel, the smooth muscle in that tissue will squeeze, and platelets around are going to release, are going to become activated. That causes a platelet plug to form. It's not the clot, but the platelets get sticky and it tries to hold to each other and to red blood cells to give a little bit of a stop. For tiny capillaries, sometimes this is enough to stop the bleeding. But if it's a big vessel, you need to form the clot, and that with the activation of the platelets and calcium and clotting factors, basically there's 2 enzymatic reactions. You see this blue netting that's over top of it? That's called fibrin, and that holds that platelet plug and anything else that comes in contact with it, sticks it, and pretty soon you get a clot. It's a lot like a scab on the outside of your skin. Okay? It just happens on the inside. So the damaged blood vessel will constrict, a platelet plug will form, and then you form a clot. That's basically the 3 steps to hemostasis. Alright, so what is a CBC? CBC stands for a complete blood count. And basically it's what a medical lab would do to test your blood for a variety of things. They're going to count your red blood cells, your white blood cells, and your platelets. They're going to measure your hemoglobin and your hematocrit. They measure a whole bunch of different things. You can see all this stuff listed here. And it helps a doctor diagnose what might be wrong with you, or at least get an idea of what might be wrong. So it helps in the diagnostic process. It's also good to know whether you're at homeostatic levels for these things. This is pretty much common lab work done every year at your physical, or once you start to do that. And usually it'll tell you whether you're high or low, and sometimes they even have information what it might mean if it's high or low. Okay, so probably some of you have had this done, or you will at some point in your life. And there is an example of one in your lab book, and you will learn a few more things about what this is measuring. But WBC is white blood cells, and 5.88 times 10 to the 9, okay, per liter. So what that means is 5.88 9 would be 5 million, right? 9 zeros. No, millions is 6. 5.88 billion per liter, or million per milliliter. And remember, you want 5 to 10,000 per mil. So sometimes the labels, you got to think about how to get that down to what's normal and what's not. Red blood cells 10 to the 12, that's going to be 4.4 billion per liter or 4.4 million per milliliter, which is close. Okay, so this guy's white blood cells seem a little high to me. So what if your CBC showed a low number of red blood cells? Why would that be bad? Okay, low red blood cells. Think about what red blood cells do. They carry oxygen and you don't have enough. Okay, so you're probably anemic. You may be tired because you're not carrying enough oxygen. If your white blood cells are high, that might indicate what? Think about what white blood cells do. Usually high white blood cells indicate an infection, particularly a bacterial infection. Okay, what about platelets? If your platelets were low, platelets are involved in clotting, so without them you might bleed more easily. Okay, I'll go back to that slide for a second there. This is a good stopping point. It's about a third of the way through the PowerPoint because this is the end of the blood section. The next section is on blood vessels. It's a little bit shorter, but this is a good time to stop if you need to take a break. Okay, so the blood vessels include the arteries, the capillaries, and the veins. Basically, arteries take blood away from the heart. They branch into smaller units called arterioles, and eventually branch into smaller tubes called capillaries. The capillaries are the smallest blood vessels, and that's where everything happens. All nutrient exchange occurs at the capillary level, because arteries and arterioles, their walls are too thick. You can't get things across. And when the capillaries merge into venules and then veins, also the walls are too thick. You don't have any nutrient exchange in the veins or venules. Eventually the veins merge into the superior and inferior vena cava and dump back into the heart. So moving from left to right along the bottom, the heart is over here on the left, the aorta to the arteries to the arterioles to the capillaries, where all nutrient exchange takes place, which merge into venules, which merge to veins, which merge to the vena cava, and back to the heart. Okay, so arteries take blood away from the heart, veins take blood to the heart. Okay, you can see this summarized in a YouTube if you want to stop and watch that video. Again, the capillaries are where all the action is. It's the smallest of the vessels. It's barely bigger than a red blood cell in diameter. Okay? And nutrients and oxygen tend to go out of the capillaries into the tissues, and carbon dioxide and other wastes that the tissues generate get pushed back into the capillaries, and away it goes to wherever it needs to be taken care of. How do things move in and out of capillaries? Via diffusion, via osmosis, which is just water, remember, and blood pressure can push things out. Okay. Diffusion is movement from high to low concentration, and this is mostly the very tiny things and the gases like oxygen. Osmosis is the movement of water, and filtration, anything can be pushed between the cells. You can kind of see that here. Blood pressure is higher at the arterial end because that's where the heart's over here, and blood pressure pushes stuff between the capillary cells out into this space so the tissues can soak it up. Okay, glucose, water, amino acids, etc. get pushed out into space because of blood pressure. And then as you get towards the venule end of the capillary, there's less blood pressure, and waste then can get put back into the capillaries and taken away to the liver or kidney to get rid of it. If blood pressure is too high, too much stuff can be pushed out, and your tissues can be swollen. Too much water gets pushed out, you get swelling or edema. That's one of the side effects of high blood pressure, and it hurts because it presses on nerves. So capillary exchange. Again, the capillary is where all the action is. This little dude shows some of the general names of the blood vessels. The list of the ones that you need to know are in your lab book. Pretty much the rule is they're named for the body region they go through, and every time they get into a new region or split, they get a new name. So if you look right here, this is the common iliac artery, and it splits right there to the internal and external iliac artery. And when it goes through and becomes over your thigh region, then it's called the femoral artery. When it goes past the knee, then it's called the tibial artery. And almost always, for every artery, you have the same vein. So this is the femoral vein, the tibial vein, the external iliac vein, internal iliac vein, and common iliac vein. So pretty much the names are, if you have a vein, you have an artery. Alright, so that's the end of the vessel part of this. The rest of this slideshow is about the heart itself. Your heart is tipped a little bit to the left, takes up more room on the left side of your body than your right, and it is covered with a membrane called the visceral pericardium, and the cavity that it sits in is lined with the parietal pericardium. Hopefully you remember from Unit 1 that parietal means it lines a cavity, and visceral means it touches the organ itself. If you're in class to dissect hearts, sometimes the pericardium has been removed and sometimes not, so you can see how thick it is, but it's protective. And then there's fat on the surface of the heart as well as an energy source. The heart is one great big muscle, cardiac muscle in particular, and it's called the myocardium. It's very thick. People eat heart because it's a muscle, but I've heard you have to soak it to get all the blood out of it because it's very well vascularized, lots of blood in it, and blood can taste kind of metallic apparently. So there you go, if you eat heart, boil it first. The lining of the heart, like everything else, lining is epithelial tissue. It's called the endocardium. So here's looking at the heart from the outside. The person that this heart is in is facing you. So this would be his or her right side. The top chambers are the atrium, right atrium, left atrium, and the bottom chambers are called the right ventricle and the left ventricle. So there it is, looking at the surface of it. Okay, and then the next picture shows you the chambers. So again, the atria are the top two chambers, that's where blood enters, and the ventricles are the bottom two chambers, and that's where blood leaves. So looking at this picture, this is going to be the right atrium and left atrium are the top two chambers, and then the right ventricle and left ventricle in the bottom. So blood enters the right atrium from the body, it enters the left atrium from the lungs. When this fills up, the valves open, and the blood, most of it, literally just falls into the right or the left ventricle. The right atrium, the blood in there dumps into the right ventricle, and the left atrium, the blood in there, dumps into the left ventricle. These two valves are the tricuspid and the bicuspid valves. Okay, you can see the white tissue there is the valves, hooked onto the bottom with these little things called chordae tendineae and the papillary muscles. But when the blood gets into the ventricles, when the heart squeezes, from the right side it's going to go through this right here to the pulmonary artery to the lungs. From the left side it goes up through this valve, the aortic valve, up through the aorta and pumped to the body. And when the heart squeezes, you don't want blood to go back up into the atria. That would be backwards. So these valves close to prevent that from happening when the heart contracts. Okay, those valves are called the aortic valve and the pulmonary valve. So the atrioventricular valves, the tricuspid on the right, the bicuspid on the left, the bicuspid is also known as the mitral valve, prevent blood from going back into the atria when the heart contracts. The papillary muscles and chordae tendineae hold those valves closed, and the pulmonary and aortic valves prevent blood from falling back into the ventricles when the heart is filling back up again. So here's the heart again with its parts. Okay, bicuspid, tricuspid, pulmonary valve, and aortic valve. Again, blood comes into the right atria from the body. This is the vena cavas here, these blue valves, and comes into the left atrium from the lungs. When they fill up, the blood pretty much falls into the ventricles, a little bit of squeeze there to get the last little bit out. And then when the heart squeezes, it squeezes down here, the apex or the tip of the heart squeezes first and pushes blood from the left side out the aorta and from the right side up the pulmonary artery. Notice the right side, the walls are much thinner than the left because the right side's only got to pump blood to the lungs. The lungs aren't very far away from the heart, right? They're right next to it. But the left side, it's got to pump blood out the aorta, which is taking blood to the top of your head and all the way to the bottom of your feet. So the left side is much stronger than the right side. All right, so if you're tracing blood, and I'll do it with the picture first here. If you were tracing blood from the vena cava to the right atrium, through the tricuspid valve, to the right ventricle, through the pulmonary valve, to the pulmonary artery, to the lung, pulmonary veins right here into the left atria, through the bicuspid valve to the left ventricle, through the aortic valve to the aorta. That would be the pathway of blood. So if you want the words, there they are. The vena cavas to the right atrium, through the tricuspid valve to the right ventricle, through the pulmonary valve to the pulmonary artery, which carries blood to the lungs. It comes back via the pulmonary veins into the left atrium, through the bicuspid valve into the left ventricle, and when the left ventricle contracts, through the aortic valve to the aorta to the body. Make sure you can trace the pathway of blood through the heart, no matter where I tell you to start. Okay? So here's another picture. The blue arrows mean the blood doesn't have any oxygen in it yet. But when it comes back from the lungs, of course, now it should have, it should have oxygen, and now the arrows are red. Okay. Okie dokie, to make this contraction happen, you have to have a nerve impulse. So there has to be electricity, right? Cardiac cells, and if you look at this cell, it looks like it's beating. You actually grow them in culture, they'll beat. Once they start to touch each other, they'll start to twitch, which is really kind of creepy when you look at it in a petri dish and they're twitching. Okay, but cardiac cells are self-excitatory. They have their own little pacemaker called the sinoatrial node, and that little phrase, you beat your own drum, is quite true. Everybody's got a pace, and some are higher than others. You can blame mom and dad, right? Some of it's genetic. And obviously things can make it speed up or slow down, the sympathetic and parasympathetic nervous system for one. Okay, but they will beat on their own. The current that makes that happen stimulates the heart to contract, can be measured. And you do that by an ECG, also sometimes called an EKG, because cardio starts with a K in German. But ECG, EKG measures the electrical conductivity of the heart. So what is being measured on the y-axis is amount of voltage, and it's comparing across time, and this is what a normal EKG might look like. Okay, the wave pattern can be used to diagnose heart problems. You can get paid to just read ECGs all day, every day. Okay, it's a pretty high-paying job, actually. Okay. What's being measured here is the voltage. These volts, or that change in electricity, is what causes the heart muscles to contract. Pretty much you go into an emergency room, they're hooking you up to this to make sure you're not having a heart attack. Alright, the next topic is cardiac output. Basically, how good is your heart? How much blood will it pump out every time it beats? Okay? If your cardiac output is higher, you don't have to beat as much to move blood around as you would if it didn't pump very much at one time. Does that make sense? Cardiac output is how much blood is pumped out every time you squeeze that heart. Okay? And it can be measured. It's kind of a fancy machine. You have to get a radioactive tracer and sit under this machine and have it take a picture of your heart every 0.1 seconds or something to estimate it. So it's kind of cool, but it's also very expensive and not measured easily without, an appointment and that type of thing. Not like an EKG, we can just hook you up and watch it happen. Okay, so why does it matter? If your cardiac output is too low, you're not going to get blood to your tissues, and your tissues are going to die without oxygen. On the other hand, if your cardiac output is really high, that could indicate high pressure, which could cause your vessels to burst, and that wouldn't be good either. It depends on two things: how fast your heart beats and how much blood is moving each time it does beat. Your heart rate we can easily measure, right? You can take your pulse. It's harder to measure stroke volume, but it depends a lot on blood volume. If you're dehydrated or you've bled out or been stabbed or have an injury, your stroke volume may go down. Okay? Because that really depends on how much blood enters the heart and how strong your heart is to pump it out the next time. Okay? So we need to know your heart rate and stroke volume, which is harder to measure. There's no gold standard or very accurate measure to get your collected information on that to calculate cardiac volume without putting you under this machine that just uses radioactive tracers. So we tend to measure heart rate and use that as an indicator of health rather than try to measure cardiac output, because we don't have a good way of easily measuring your stroke volume. Obviously, we know stroke volume is going to go down if you don't have enough fluid. But other than that, it's harder to measure. So, heart rate, what's it supposed to be? You guys remember? About 72 beats per minute. It tends to go up with age, fitness levels, whether you're male or female. Some hormones can mess with it. Obviously, epinephrine, your heart rate's going to be higher. Whether the parasympathetic or sympathetic nervous system is activated, whether you're nervous or not, whether you had an espresso that morning, for example. Okay. Stroke volume depends mostly on the amount of blood, so blood loss, sweating, drinking a lot of fluid can change this, but it's not easily measured. So what happens to heart and your cardiac output if you drink an espresso? Okay? Or you see a deer run in front of you. Both of this is going to increase your heart rate, which will increase cardiac output. But what if you meditate or relax or do something to calm yourself? This will probably lower your heart rate, which will lower your cardiac output. Okay? What about exercise? What does exercise do to heart rate? Okay? Again, tends to go up. Okay? I am going to fix that for a second because this should say heart rate. Okay, put this back up here for you guys. There you go. Alright, the last thing we're going to talk about, or one of the last things in this PowerPoint, is blood pressure. Remember what blood pressure is supposed to be? Okay, 120/80, because I'm sure you just came up with that right at the top of your head, right? Everybody remembers that? Blood pressure depends on four things: your heart rate, stroke volume, so your cardiac output, peripheral resistance, which is essentially the diameter of your blood vessels. Vasoconstriction increases peripheral resistance, and vasodilation, when the vessels relax, decrease peripheral resistance. Blood volume, how much blood you have, and blood viscosity, how thick your blood is. If any one of these things goes up, blood pressure goes up. Okay? Any one of these goes up, blood pressure goes up. So if you drink a lot of fluid, your blood volume goes up, your blood pressure goes up. If your heart rate goes up because you're scared, blood pressure goes up. If vasoconstriction happens, your vessels get tinier, it's harder to push blood through, increases your blood pressure. If your blood gets thicker, if you have high serum triglycerides because all you eat is McDonald's French fries and milkshakes, makes your blood viscosity go up, makes your blood pressure go up. The opposite is also true. If any one of these four go down, blood pressure will go down. Okay, so try to remember these four things are the main regulators of blood pressure. Blood pressure, when it's high, is diagnosed as hypertension. You have high pressure. Okay, why is it dangerous? Well, if you put more pressure on your hoses, the hoses could burst. That would be a stroke. Okay, and if blood vessels burst, they're not carrying oxygen to the tissue. Okay, and they could die. One of the first things that happens when the high blood pressure is the tiny capillaries in your eyes burst, and it can cause, when they heal, blind spots. So eye doctors can know if you have high blood pressure by looking in the back of your retina and seeing if your capillaries have burst and healed. Okay? High blood pressure pushes more fluid into the tissues, and that causes swelling and edema, and that can cause pain because it's pressing on nerves. Okay? What makes you more likely candidate to have high blood pressure? What are the risk factors? A risk factor is something that makes you more likely to have hypertension or have whatever it is that they're talking about. What makes you more likely to have high blood pressure? Being overweight, okay, is probably the number one reason. Not exercising. Males have a higher risk than women until menopause, and then it's about equal. Smoking. What else? There's always a genetic component. Okay, so there's lots of things that make you at risk for high blood pressure, but diabetes and obesity tend to be the biggest problems in the United States. Okay, it's supposed to be 120 over 80, so if you're at a doctor's office and you are 140 over 90, you're going to be diagnosed with high blood pressure. Basically now they're saying anything with the top number above 135 or the bottom number 90 or higher, you are at risk or you are likely to be diagnosed with hypertension. Okay, so a blood pressure cuff is the most common way to look at blood pressure and then be diagnosed with high blood pressure. All kinds of medications are used to treat this. Remember the four regulators? We have drugs that will target all of those. If your blood's too thick, you have high blood viscosity, they might give you a blood thinner, for example. If you have high blood volume, they might give you a diuretic so you pee more. Reduce your blood volume, reduce your blood pressure. If you have vasoconstriction problems, they might give you a vasodilator like nitroglycerin. If you have a high heart rate, they might give you a calcium channel blocker or a beta blocker. It blocks epinephrine, for example. So there's lots of different ways to treat high blood pressure. And hopefully they tell you some lifestyle things like go home and lose weight and stop eating French fries and chocolate shakes. Okay. Your body will do almost everything it can do to try to lower blood pressure as well. If one of those four things go up, one of the other three will go down to try to compensate. Although generally viscosity is not a compensator. You can't make your blood thinner quick enough to react. So for example, if you were to lose a lot of blood, you got hurt, injured, cut yourself, whatever, then you're going to drop your blood pressure because your blood volume will go down. Immediately there's going to be constriction of your blood vessels, especially in the area where your injury is. And that increases peripheral resistance, which will make your blood pressure go up. You can also increase your heart rate, and that will make your blood pressure go up. So the initial problem, the drop in blood pressure because of blood volume was lost, can sometimes be compensated for by one of the other three going the opposite direction. Okay, so heart rate and peripheral resistance, vasoconstriction will increase to try to get your blood pressure back to normal. So that will of course go on, but sometimes people with hypertension need further help than what their body can do. So medications are used. But the whole goal, again, is to get homeostasis back to normal. So what if you got drunk? Okay, lots of people do this. They drink and drink and drink. What's going to happen to your blood pressure? Well, before you pee, all that extra fluid is going to make it a little harder for your your heart to pump around, because some of that fluid gets into your blood. And so now you're pumping around 6 liters instead of 5. That will increase your blood pressure. Okay, that increase in blood volume increases blood pressure. So what are you going to do? Your body will lower heart rate and vasodilate to try to compensate. And that does happen. When you drink alcohol, your heart rate goes down, your peripheral resistance goes down, you might look even redder, okay, because of vasodilation. But both of those things will help compensate for that extra fluid. Now later, of course, even though this will lower your blood pressure, you're going to pee, right? You're going to get rid of that liquid, and that will bring blood pressure back to normal. Okay, so later your urinary system will increase output, further helping your blood pressure go back down. Okay, so it's not like our body can't deal with this kind of thing, although repeatedly doing this could cause some damage as well, as you might guess. Okay, but your body will try to compensate. Again, the goal is to maintain homeostasis. Your body will do just about anything to do that. So, how can you help yourself? Get in better shape, eat healthier, exercise, exercise, exercise. And, you know, I say that and I don't exercise enough, but this is really the #1 way to help improve your heart health, is just simply move around. There are medications to help with high blood pressure. I think we talked about some of these. Diuretics to make you pee, to lower blood volume, block the sympathetic nervous system. Beta blockers and calcium channel blockers work this way. It basically makes your heart not contract as much. Vasodilators reduces peripheral resistance, and blood thinners are also used to help lower blood pressure. There's bypass surgery you can use to fix a coronary blockage, and I'll show you a picture of that here. And angioplasty is another technique used to fix blockages. Your heart is a muscle, and if blood vessels are blocked to that tissue, those cells are going to die because they don't get enough oxygen. And cardiac cells will not regenerate. So once they're dead, they're gone. So when they find like this right here, a blockage, they know that the tissue beyond that point sometimes is going to run out of oxygen or be damaged. So they'll take a little piece of a vein from your leg and they'll create this bypass, like a road around the blockage, so this tissue now, blood flow is restored. And when you hear somebody had a triple bypass, they had 3 such bypasses put in at the same time. I think they do quintuple bypasses. I think they've done 5 in one heart in one operation before. Okay, but bypass surgery is used when there's a blockage, usually because of high cholesterol or something else, in a vessel that's in the heart muscle itself. An angioplasty, a balloon angioplasty, is here's a vessel in the heart, okay, that cholesterol has made it so the blood flow is barely there. They can put in this balloon that's wrapped with this little wire mesh, and they get to the place where the blockage is, and they blow up the balloon, and so that pushes the vessel open, and when they pop the balloon or deflate the balloon, the mesh stays there, and it holds that vessel open, restoring blood flow. So now the tissues beyond that point in the heart can get more blood, which means they get more oxygen. So angioplasty, as well as bypass surgery, which we just did on this slide, are 2 ways they can help restore blood flow to the heart. So that is the end of Unit 10, and there's a lot of information in this, so you might want to listen to this twice, or take good notes, make yourself flashcards, do whatever you need to do. Practice with the Quizlets, check out your lab manual, and as always, you can email me if you have any questions.